And turning to tonight's room, I'm delighted to introduce Distinguished Professor Ian Paulsen, who's a Fellow of the Society, a Fellow of the Academy of Science, Fellow of the Academy of SM. Ian? Microbiology Society from Macquarie University in Sydney. You can see a lot of Ian's credentials on this slide. He is Director of the ARC Centre of Excellence in Synthetic Biology and Director of one of Australia's national research infrastructure facilities, the Australian Genome Foundry. Tonight he's going to synthesise 30 years of his research in synthetic biology, a field of research in which he's a global leader, and his title, because of his deep knowledge about the subject, is Inspired by Nature, Designed by Science. And I'm sure that you'll enjoy his ability to draw a big picture about synthetic biology for us. I've put a few of his credentials on this slide, but I add to it in 2014, Thomson Reuters identified him as one of the world's 3,000 most influential scientific minds. In 2020, he was named one of Australia's research superstars in an annual list of the top 40 researchers considered to have made a major contribution to science during their career. And in 2024, he was awarded the New South Wales Premier's Prize for Science and Engineering in the category of Excellence in Biological Sciences. His success, of course, is based on a deep understanding of his field of research gained over 3 decades. They started when he undertook his PhD in microbiology, from which he graduated in 1994, which is where I started my 30 years. And then he spent 12 years in the US, first as a postdoc at the University of California, San Diego, and then for at least 8 years as faculty member of the Institute for Genomic Research, known as TIGA, in Rockville, Maryland, where he led many microbial genome sequencing projects. He returned to Australia, to Macquarie University in 2007, and the rest is history. Ian, welcome to the lectern.
Well, thank you for the very kind introduction and for the opportunity to talk to you tonight. Where am I going to put my props? I'll leave them there. Okay, so I haven't spent 30 years as a synthetic biologist because I only got into synthetic biology 10 years ago when I decided there was this really cool new area of science called synthetic biology, and after plotting with my DVCR and friend Saki Pretorius, we decided to make a big strategic push into synthetic biology, and here I am. Okay, so I'm a microbiologist. I don't understand anything bigger than a single cell, and I certainly don't understand people. When people think of microbes, you usually think of them as causing disease and stuff like that. Maybe it's now, people are starting to realise now that your gut microbiome, the microbes in your gut are important for your health. I'm here to tell you that there would be no life on this planet without microbes. So what can I tell you about microbes to start with? They're really tiny. Okay, so what do we know about microbes? They're really small. How small, do you ask? Here we have my fluffy stuffed version of E. coli, 1 million times life size. E. coli is essentially the lab rat of microbiology. This is yeast. It claims to also be 1 million times life size. That must be a lie because it's actually about 10 times bigger than E. coli. But we'll go with that for the moment. Yeast, of course, gives us brewing, winemaking, baking, so responsible for a large chunk of human happiness and maybe misery. What else have I got in my box of tricks? Anyone want to guess why this one's wearing a cape? It's a superbug. That's golden staph, highly multi-drug resistant bug. So this is one of the pathogens. I've got a huge collection, although I didn't bring them all. So why am I showing you microbes? Is just give you an idea of their scale, and I want to tell you that there is a ridiculously large number of them. So there's 5 million, estimated 5 million trillion, or 5 followed by 30 zeros, microbial cells on Earth. You actually have as many microbial cells living in you and on you as you have human cells. So arguably you are vehicles for transporting and feeding your microbial masters. And over the course of 3.8 billion years of evolution, microbes have adapted to survive in essentially and flourish in every environment on Earth. And that ranges from boiling volcanic pools in the top left there. There's a photo there of microbial biofilms growing at the bottom of a lake in Antarctica that's frozen on the top. There's another lake there, Mono Lake, that's insanely salty and is highly alkaline. They even grow in new environments that humans have developed. You get microbes that grow inside nuclear power plants. And so microbes over the course of this evolution have evolved a huge diversity of function, both in terms of the genes they carry and in terms of the functions they can carry out. And it's that tremendous diversity of function that synthetic biology tries to take advantage of. Oh, it's just slow. Okay, all good. So what is synthetic biology? In synthetic biology, we sort of try and apply an engineering mindset to make engineering biology easier. What does that actually mean? We sort of treat genes and DNA as if they were little Lego blocks that we can put in different combinations. So I can, for instance, take a gene or two from Staphylococcus aureus, a gene or two from E. coli, put them into yeast and try and make yeast do something that it doesn't naturally do, produce some economically useful compound, produce some pharmaceutical, do something that's useful for humanity. And inspired by engineering, synthetic biology has this iterative design, build, test, learn cycle. What does that involve? So in design, we'll sit down on a computer and decide what genes from what organisms we can take and put in some new combination that allows some microbe to do something it doesn't naturally do. In the build phase then, we actually synthesize the DNA and we put it into the new organism. And then because biology is complex and we don't understand 90% of it, we then have to test to see if our idea works. And I'm here to tell you that most of the time it doesn't. So we test it, work out why it doesn't work. Maybe the gene I took from this organism doesn't like to work in yeast, so I've got to get another alternative from a different organism. And we test to see what goes wrong, and we then learn from that, redesign, rebuild, retest, relearn, and keep going round and round that cycle until we successfully engineer our organism to do what we want it to do, or we decide it's too hard and we give up and we do something else. And in case you're wondering why there's a QR code there, that if my explanation is total confusing murk, click on the, get that QR code, and there'll be an animated explainer video made by my centre about what synthetic biology is. So in essence, synthetic biology is a platform technology, and we can apply it to trying to solve many different problems that faces Australia and the world. So we can try and engineer our microbes to produce proteins as new diagnostics, to produce new therapeutics, to improve agriculture. Very frequently, one of the main drivers is trying to produce, let's go back. Trying to produce, no, the laser pointer doesn't want to work. Trying to produce chemicals, particularly what we'd like to do is replace many of the chemicals that we currently get from fossil fuel refining. Can we replace them with synthetic biology to get microbes to make sustainably produced alternatives to the chemicals and plastics and that we get from the fossil fuel industry? We can also try and engineer them to clean up the environment, to bioremediation, to produce energy in the form of hydrogen gas or other fuels, and also to produce novel biomaterials with novel capabilities, novel features. So one of the things I'm director of is the RSC Centre of Excellence in Synthetic Biology. So that's a 7-year, $50 million research centre. We're now at the end of year 4, so we're roughly past the halfway point. And every ARC Centre of Excellence has to have a vision of how it's going to change Australia and the world to make it a better place. In our case, we're trying to engineer our synthetic microbes to take either agricultural biomass or to take waste streams from industry or agriculture or elsewhere and turn them into higher value products, pharmaceuticals, platform biochemicals, fuels, biomaterials, plastics, etc. And so within the Centre of Excellence, we have a core team of synthetic biologists who are trying to engineer microbes and are doing various science fictional things like trying to engineer microbes to do stuff together that they can't do by themselves, or trying to engineer new organelles into the microbes, or in neobiochemistry, trying to produce new to nature enzymes or pathways. But in order to have an impact in the real world, the centre also includes a range of other science and humanities and engineering researchers. So in our industrial translation capability, we have a team with expertise in fermentation, in chemical engineering, in techno-economics, in industry strategy and policy, who study how and why industry translation works in one place or not another place. Or to put it another way, how can we influence government policy and industry strategy to enhance translation of these technologies in Australia? And in our social dimensions team, we have a team of researchers with expertise in ethics, in technology law, in art and design, in sociology, and who are studying these sort of social dimensions and the social impact and the social acceptance of synthetic biology. So the Centre of Excellence, every Centre of Excellence is like a massive octopus with a tentacle everywhere. We have 17 research groups across 9 universities, so about 250 researchers in total. This is from one of our annual conferences. And we have, to date, I think now 35 industry partners or affiliates. And I think over the course of 4 years, we've published 40 to 50 papers in journals that have science, cell or nature in the name. So we'd like to think we're doing exciting science, but one of the things the Centre of Excellence is also trying to do is have impact in the real world. So I'm super proud to be able to say that in 4 years we've spun out 9 startup companies that have raised $200 million in venture capital. So I'd say that if there's anyone in government in the room, I'll say that's a 10-to-1 return on the government investment so far in the centre. And I'm going to walk through each of these, not as an advertising piece, but to actually give you an idea of the range of different things one can do with synthetic biology. Full disclosure, I own a tiny shareholding stake in 4 or 5 of these companies. So if any of them become a $1 billion company, I may not have to eat cat food in my old age. But I'm not, the odds of that are relatively low. But let me show you, again from my bag of tricks, let me show you some products. I'm going to start with this. So in fact, maybe I'll even wear it. I was going to say, I'm also super proud to say 7 of these 9 startups, the founders are either PhD students or postdocs. So they're young early career researchers. And if there's anyone from the New South Wales state government here, I would point out that all 9 of these are based either in New South Wales or the ACT. Anyway, propaganda aside, or maybe it's not aside, so I'm going to talk about some of the companies. So New Era Bio was founded by one of my postdocs and actually one of Amy Kane's postdocs who's in the audience tonight. And so they are making dyes, fabric dyes, textile dyes. Most textile dyes are chemically based and production of them leads to huge waste problems. So they're making sustainably produced, safe, colour fast fabric dyes from bacterial pigments. And this is just a silk piece of silk dyed by one of them. Another company is Number 8 Bio, spun out by one of my postdocs and one of my PhD students. They have engineered yeast. This is some spray-dried yeast, in case you don't know what yeast is. I can probably actually pass that around. So every little tiny particle in here is millions of yeast cells that have been dried down. And it looks to be spilling everywhere here. Sorry, I'm making a mess. Don't shake my props, apparently. So Number 8 Bio are making synthetic yeast that can be put in a cattle feed and stop cattle producing methane. They actually have now found a non-GM way to duplicate what they made with their GM yeast, and so they've now done nearly a dozen cattle trials with their first product and show 88% methane mitigation and a 12% increase in cow weight on the same amount of feed. So they're going to market with their first product this year and are hoping that they've got both an economic product which will help farmers but will also substantially mitigate methane production, a important greenhouse gas driving climate change. And then I think I've got one more. So Laveur is another startup company out of the centre, spun out by one of my PhD students, Tom Collier. Tom and his wife are passionately interested about stopping deforestation in Southeast Asia, and so even before in fact he hasn't submitted his PhD yet, and I'm not sure when or if he will, but he's founded a successful startup company which is using yeast fermentation to produce oils to replace oils that industry uses from palm oil plantations, which are one of the drivers of deforestation in Southeast Asia. And so he's now been successful in raising significant venture capital to try to engineer yeast for production of particular oils that can be palm oil substitutes. From ones that I don't have props, so Samsara Eco, they're actually the furthest along in their journey. They've just raised $100 million to produce their first full-scale plant. They do plastic recycling. They've produced a very efficient enzyme that can recycle the plastic in plastic drink bottles into the original monomer, and that can then be used for making new plastics rather than having to make them from fossil fuels. I'm not going to talk about Curion Diagnostics because it's sort of non-functional now. Swan Genomics, spun out from Lawrence Lee's group at UNSW. They've built a DNA origami protein structure, and here I'm going to use words that I don't understand, that perfectly positions DNA polymerase and then can use surface plasmon resonance, they're the words I don't understand, to dramatically increase fluorescence, and are developing a single molecule DNA sequencing device out of it, and have received significant, they've brought on co-founders from the US and received significant US venture capital to do that. Wild Year Zoo, partly from my and Amy's group, but mostly from the University of Newcastle. This actually started out of a citizen science project that then turned into a startup company where they had people going out into their backyards and something and collecting native yeasts, and they've been isolating yeast with weird capabilities from the environment and characterising them and finding some with novel industrial utility. Entozyme is founded by an academic at Macquarie, Maciek Masalko. So Maciek, I engineer bugs. Maciek also engineers bugs, but he engineers different sort of bugs. He engineers insects. So he's engineering black soldier flies. Black soldier flies can eat essentially sewage or any organic waste stream and turn it into insect biomass. He's seeing if he can engineer the black soldier flies to, instead of just producing biomass that can be used for aquaculture or animal feeds, he's seeing if you can produce much higher value products such as enzymes for laundry detergents and things like that. And then lastly, Biomega is engineering yeast strains, and that's at least mildly connected to Macquarie, but also University of Western Sydney, engineering yeast strains for production of omega fatty acids and also other nutraceuticals. So hopefully that gives you a very quick and eclectic overview of the types of and the wide variety of industries that synthetic biology can conceivably have an impact in, and also an example of how we're trying to have an impact in Australia and the world. I am a big gaming nerd. I have a severe gaming addiction. when I go home to relax, I play computer games or Xbox games. So I'm super excited that the young researchers in my centre have developed an educational card game, Remediate, where you play as a microbe and you try and detoxify the environment. And these are images from various public events we've run around Australia. So we've run public events with this game around Australia, and we're currently working with 2 New South Wales schools to develop an entire curriculum around this. And like I said, I'm a gaming nerd. I've played this game. It is a fun game to play, and along the way you can accidentally learn stuff. And I'm going to say, and again, I give credit to this one to Amy, who's here. We've also run events at places like the Woodford Folk Festival, where we've run panels on what synthetic biology is, and try to lure some of the 140,000 people there away from music and learn some science. We also have made a series of animated videos on things like what's synthetic biology, what are biosensors, how do you actually molecularly change an organism, how do you use gas fermentation to make aviation fuel, how do you use precision fermentation, what are all these things? And this QR code takes to a landing pad with all those videos. And we're also trying to impact policy and in Australia, so we've produced various policy pieces for state, federal governments, for academic journals and non-academic publications. And last year, let me throw my microbes around. Last year, we ran a Parliamentary Friends of Science event in Canberra, chaired by Ed Husick and Richard Marles, the Deputy Prime Minister, where we had 3 members of our centre on a panel there explaining synthetic biology to politicians and staffers in Canberra, and hopefully getting support for the bioeconomy on the Australian government's radar screen. The other entity that I'm director of is the Australian Genome Foundry. So this is a robotic automated facility to try and accelerate synthetic biology research in academic and industry research in Australia. So I'm going to say for 50 years, molecular biology has been a cottage industry where you have people with decades of training, essentially skilled artisans, sitting at a lab bench doing repetitive tasks over and over again, pipetting from one tube to another, things like that. But I'm trying to bring molecular science into the industrial era, and can we have robots do the boring repetitive tasks and free up humans to do the creative thinking? Or in other words, can we get robots to do stuff 100 times faster than people can do it? And this is our lab here. I'm going to say the robots on the right and the people on the left, roughly speaking. And so I think this basically says what I was just trying to say. So instead of having one person pipette one thing at a time, let's have a robot do it 100 times in parallel. So instead of building one synthetic microbe at a time, we can build 100 at a time. And if you think back to the design, build, test, learn cycle I talked about, because usually our designs don't work the first time, if you're making one design at a time, you've got a small chance of success. If we can make 100 designs at a time, then we've got 100 shots on goal rather than one shot on goal. So the Australian Genome Foundry offers a variety of services to academic and industry clients. We can build synthetic microbes. We can do high throughput in fermentation under industrial conditions, small scale ferments. We don't have 1,000-litre tanks or 1,000,000-litre tanks. But we can mimic those at small scale and high throughput. We have high throughput analytical chemistry to test for what our microbes are making. I don't want to talk about flow sorting and flow cytometry. And then importantly, we actually incubate companies directly in the foundry. So I've had 6 companies incubate in the foundry so far. 4 of them are still currently there. Some of them are startups out of the Centre of Excellence, but some are companies that have actually moved to New South Wales in order to incubate at the foundry and get access to the foundry capabilities. So this is just a bunch of videos showing some of our robots in action. So a common thing you do in synthetic biology is transformation. That's basically getting a piece of DNA into one of our microbes. And normally a person would sit there pipetting stuff at a bench for a few hours to do that, doing one transformation at a time. These are robots doing 100 transformations in parallel in a fraction of the time. And we're actually also reducing our plastic weight. If a person did 100 transformations, they'd plate it onto 100 plates. We can do 100 transformations on one plate. So the Foundry is trying to do things quicker and faster just to accelerate the pace of synthetic biology research. The Foundry is essentially, so we're funded by NCRIS, and I never get this acronym right. Let's see if I can get it right tonight. The National Cooperative Research Infrastructure Strategy. That may actually be right. Funded via Bioplatforms Australia, and we do fee-for-service work, we do collaborations, and we have companies incubate. To date, we've been operational for about 12 months now. I think to date we've had about 12 to 15 industry clients, 20 or 30 academic clients that no longer fit on one slide. But our driver here is not to make, yes, we don't have to charge full cost recovery. Our NCRIS and Bioplatform masters don't want us to charge full cost recovery. We're there to try and accelerate and help research. And so I'd like to think we're starting to build up, I'll call it a bioeconomy ecosystem in Australia. And so if we think of going from left to right on this slide along those arrows, if we think of this maybe as the journey of a startup company, then you get ideation from cool science, from out of the Centre of Excellence or out of other research across Australia, for instance, out of the Advanced Engineering Biology Future Science platform at CSIRO. These companies then the founders are often scientists who know nothing about business, and so they can go to startup accelerators and most of our startup companies have gone to the UNSW SynBio 10X Accelerator or to the CSIRO On Prime On Accelerate program to actually learn what's a startup, what's customer discovery, things like that. And then where the biofoundries sit in this puzzle is that they can accelerate the journey of these startup companies. And so we have the Australian Genome Foundry and have a sister facility up at Queensland run by my friend and colleague Esteban Marcellin that does some complementary stuff. And so, for instance, the Chief Executive Officer of Number 8 Bio is on record as saying the only reason they were able to make their targets for the latest round of fundraising is because by being able to incubate directly in the Foundry and getting direct access to the equipment, they could do things. But instead of having 6 weeks turnaround for everything they wanted to do, they could have 1 day turnaround. Then, once the biofoundry is essentially there to prove things at laboratory scale, once to get out of laboratory scale and get towards doing this in the real world, you need to go to, let's say, 1,000 litre scale if you're doing fermentations. And there's both academic and industry facilities in Australia, Cauldron, at ETH, at the Macquarie pilot plant run by QUT, that one can do pilot scale fermentation. And then the right-hand part of my slide is more in my imagination. We're not really quite there yet. You have to squint a bit, but we're starting to get towards the point where these startup companies are starting to do production scale in their own facilities. So I should have put a timer on so I know how long I'm okay. All right, I'm going to talk about 2 actual projects to show you some actual science. So one of the major drivers of innovation during the history of life on Earth is symbiosis. So there'd be no plants and there'd be no animals and no humans without symbiosis. So all eukaryotes derive from some ancestral organism and that wound up with an alpha proteobacteria, a bacteria living inside it that eventually became the mitochondria. All plants are derived from some subsequent ancestor that swallowed a cyanobacteria that eventually became the chloroplast. So these are bacteria that got engulfed by some early proto-eukaryotic cells and developed these new functionalities. All our knowledge of these symbiosis events is then studying it. These events that occurred 2.7 billion and 1.6 billion years ago is by studying their current genomes and trying to make sense of what we see today. We don't really understand what happens in the early stages of a newly developing symbiosis. And so Brada Lorente in my team wanted to see, could we engineer a system to force organisms to live in symbiosis with each other and then watch how they adapt to each other over time and understand how organisms adapt in a symbiosis. And so they developed a system where they had a protein that gave resistance to a particular drug that could kill both bacteria and eukaryotes. And they split that protein in half. And they put half in one organism and half in another organism. So those organisms could only live if they were both there and the antibiotic was there, the drug was there. And on the right of this slide, in pink and blue, you can see this in yeast, where we've got one yeast strain expressing one half the protein and it's not growing in this microfluidic well. In the next one, we've got two in the blue. We've got the other half of the protein being expressed in yeast, and again, it can't grow. And then the third one, we've got one yeast strain producing one half of the protein, the other yeast strain producing the other half, and they can grow when they're together. But what we really want to do is make an organism live inside another, force a bacterium to live inside a eukaryote, and in essence, we're trying to recreate the evolution of the chloroplast. So in this case, we're taking a photosynthetic bacterium, a Cyanobacterium synechocystis, and we're trying to make it, force it to live inside an amoeba, Dictyostelium. And this system has two things we can take advantage of. Cyanobacteria produce chlorophyll, which is fluorescent, so we can see if the cyanobacteria get inside the Dicty cells by looking for chlorophyll fluorescence, which you can see here in pinky red. And Dicty is an amoeboid. It's sort of all slimy and movie, so it's amoeboid and it moves if it's alive, and if it's dead, it's spherical and doesn't move. And so here's a positive control. So here we're expressing the full-length resistance protein, and the Dicty is alive regardless of whether it's got cyanobacteria inside it or not. In the presence of the drug, it's alive because we're expressing the full-length protein. Here, we're expressing half of the protein in cyanobacteria, and actually the same half in Dicty. And here, the cells are round, they're not moving, they're dead, regardless of whether the cyanobacteria are there or not. So that's the negative control. And here is our system actually working. So here we have half of the protein being produced in the Dicty, half of the protein being produced in the cyanobacteria. The Dicty that doesn't have the cyanobacteria, there's no red fluorescence, is round and dead, not moving. And over here, alive and amoeboid and motile, is our cyanobacteria living inside a Dicty that were forced, and they can't survive without each other. This is cool. This is, I believe, the first forced photoendosymbiosis anyone's ever constructed, so we think this is the world first. However, it's not perfect. They live for a week or two. They don't divide. So Dictyostelium actually eats bacteria. We thought it wouldn't. It's supposed to not eat cyanobacteria, but we think over time it eventually eats the cyanobacteria after a week or two, and then that's a problem because then everything dies. So yeah, we still have we've got a cool system, but it's got a long way to go before we can, routinely grow up tons of Dictyostelium and cyanobacteria and recreate a land plant and see what happens. But we'd then like to use this system to study how these two organisms adapt to each other over time, and look at what genetic or other changes they make to adapt to each other over time. The second story I want to tell involves yeast, which is actually where I do most of my work these days. Why are you interested in yeast? It's been domesticated for more than 5,000 years. I like to tell my cats it's been domesticated longer than they have. To which, of course, they reply, what are you talking about? We're not domesticated at all, we're domesticated humans. It's fundamental to baking, brewing and winemaking, so arguably done more for human happiness than any other organism on Earth. It's our best studied model eukaryotic organism, and we know how to grow it because from brewing and winemaking, we know how to grow it in enormous vats, up to 10 million litres, so we can now use it, as of today, we use it for producing the ethanol you have in your E10 fuels you put in your cars is all made from yeast fermentation. But with synthetic biology, we can engineer yeast to make a plethora of other compounds and, again, grow it up in enormous tanks to do that. And so this is by no means all the wide variety of things they can do, but currently people at various places in the world have engineered yeast to make anything from oils, foods, flavourings, aromas, plastic precursors, oils, lubricants, fuels, a diverse range of biochemicals. One problem with yeast, one limitation with yeast. Yeast is like a 5-year-old kid. Or actually, it's probably like me still as an adult. It loves sugary, sweet things. It basically only wants to eat sugars, and to get sugars, we have to grow up agricultural crops, sugarcane, barley, wheat. And so that's not a great, if we want to replace everything that we get from fossil fuels, we'd have to use the entire agricultural production of the earth and then some in order to do that. So it'd be better if we can get yeast to eat waste. It doesn't like to do that, it likes sweets. We've come up with a new strategy where we want to engineer other organisms to grow symbiotically with yeast and provide things to yeast, get these other organisms to grow on waste, and then turn that waste into things yeast likes to eat. One of the organisms we've looked at is C. necator. C. necator is sort of cool. It can grow on CO2. It can grow on. It's a very metabolically diverse organism, and we're building on work from Pam Silver's group at Harvard, where they've engineered it to produce the sugar sucrose and secrete it. And so this is work of one of my postdocs, Paige Erf, and we're actually doing parallel work. We're trying this in C. necator, but we're doing parallel work in a cyanobacterium as well, with one of my other postdocs, Alicia. So C. necator can use CO2 or formate, which is an easy to use C1 compound that we can use as a proxy for CO2. We've engineered it to produce the sugar sucrose and secrete that sucrose. Yeast loves to grow on sucrose, but it grows on in a way that we don't like. The way it grows on sucrose is it secretes an enzyme out of the cell and breaks that sucrose down into glucose and fructose and then takes those in. That's no good to us because C. necator will like to eat everything. It will eat the glucose and fructose. So we've had to re-engineer yeast, and so we've made yeast so it actually, the enzyme that normally breaks down sucrose, we've made it so it can't get out of the cell, and we've put a transporter that will bring the sucrose into the cell across the yeast cell membrane. And so we've got this part of the system working. We can grow C. necator, it can make enough sucrose that yeast can grow on it. What we'd like to do, and we've engineered yeast to overproduce a particular amino acid, tryptophan, and secrete that. What we haven't done yet, it's still a work in progress, and engineering C. necator is a pain in the arse, so we may actually switch all of this work to cyanobacteria, but we're trying to engineer C. necator to not be able to make its own tryptophan, so then it's dependent on the yeast. So then the whole vision then is C. necator or cyanobacteria can grow on CO2 from the atmosphere, turn to sucrose, feed the yeast. The yeast, of course, can then make our high-value product of choice, and simultaneously, the yeast is producing a compound tryptophan that C. necator requires, so then we've got this two-way syntrophy or symbiosis between them that depend on each other. We actually are plotting to expand this to a third organism, so we're working on a nitrogen-fixing bacteria that can turn nitrogen in the air into ammonia and secrete that ammonia, which yeast can then use as an ammonium source. And we've already engineered, we've already shown that yeast can we can grow yeast and azotobacter in a co-culture without any other added nitrogen, and azotobacter secretes enough nitrogen, secretes enough ammonia for the yeast to live. And we've engineered yeast to produce uracil and excrete it and make a mutant of azotobacter that can't make its own uracil. So we're close to making this three-way symbiosis where we could have two organisms take CO2 from the air, nitrogen gas from the air, turn that into things that yeast like to live on, and then hopefully grow yeast not on sugar made from sugarcane or whatever, but from CO2 we've pulled from the atmosphere. Whether we can ever do that on scale is a different question, but we'll hopefully get it. How am I going for time? Do I have a few minutes left, or am I done? All right. Our one other strategy to try and get to wean yeast off sugars, and this is I think the first published work I've actually talked about tonight. So we're interested in getting yeast to grow on methanol. So methane is a greenhouse gas. It's also the natural gas you have in your stoves and whatever, and is also abundantly produced in landfills or from biomass in general. So we know it's going to be very hard to get yeast to grow on methane. Not many organisms grow on methane. It's very hard to grow on methane. Some bacteria can, but there are yeasts that can grow on methanol. So we know how to, and there's ways to chemically or engineeringly wise convert methane into methanol, chemical engineering wise. So we're interested, could we make yeast grow on methanol rather than as an alternative to sugars? And so we took three different, again, this is going to our design, build, test, learn. Usually it doesn't work the first time, so we tried to have three different shots on goal. We engineered three different pathways: a bacterial pathway for using methanol, a yeast-like pathway, or a Pichia-like pathway for using methanol, and a sort of combination, and put them all into yeast, and they all worked. Well, actually, the two of them worked moderately well, and we could get yeast to grow on methanol if we gave it a little bit of additional yeast extract. We've done further work to try and optimise this. The short answer is we can get yeast to grow on methanol, but it's sort of sickly and feeble and not really happy. So this has been an interesting academic project, but we think this probably has limited industrial utility in the foreseeable future because the yeast is really not happy growing on methanol. But you don't know until you try stuff. So I think synthetic biology is a platform technology which will, over the course of the next century, impact most industries on the planet. And I don't think it's a magic bullet for solving climate change, but I think any solution for climate change is going to, at least in part, involve synthetic biology. To try and accelerate synthetic biology research in Australia, we've developed this novel high throughput facility, the Australian Genome Foundry. And I talked about two projects I've undertaken within my team in the Centre of Excellence. So I've engineered this split protein system to try and force a bacteria and an amoeba to live in a symbiosis with each other. And I talked about trying to engineer yeast and either carbon fixing and nitrogen fixing bacteria, again, to live in a symbiosis together so that they could cross-feed each other. I don't actually know how to do anything in a lab anymore, so these are the people who do all the actual work. The top photo is the team from the Australian Genome Foundry, and then the bottom photo is our Macquarie University synthetic biology team. And I wouldn't be here talking about science without support from the university, from the New South Wales State Government, from the ARC, the NHMRC, and NCRIS and Bioplatforms Australia.
I've worked in this field. I've got a million questions, but I'm going to hand to the audience first. So we have two microphones, hands up with questions. Don.
Thanks for an extremely interesting talk. I'm a chemical engineer and I've been interested in synthesizing things like biofuels for a long time. And one of the problems has been that in competition with big industrial processes like the oil industry, biological pathways generally are cost incompetitive. Do you see that changing?
100% agreed. In fact, we had a conversation about this earlier. So for bulk fuels, very hard. You know, the oil industry has had 150 years, God knows how many hundreds of billions, trillions of dollars of investment, have the infrastructure, the expertise. It's a smoothly running machine. Unless there's dramatic changes in the oil price, it's really hard to compete on cost for bulk fuels. And I don't think synthetic... And if you sit down and do a calculation of how much yeast or whatever you'd have to grow to replace all the fuels for cars on the planet, it's not going to happen. Where I think synthetic biology... But we depend on fossil fuels for not just fuels for our cars or planes or whatever. We depend on it for a diverse range of plastics, lubricants, emollients, a vast array of platform biochemicals. That's the space where I see synthetic biology being able to have an impact. Let me give one example. So I'm going to try and get the chemical name right. I may get a number wrong. 1,3-butanediol is an important platform biochemical. There's a process being produced that a particular company has engineered E. coli to produce it and is now producing at scale. So currently 50 million tonnes a year of 1,3-butanediol are being produced by yeast fermentation, and there's plants under construction as of today that would enlarge that to 300 million tonnes a year. So I think that's, in terms of replacing stuff from fossil fuels, bulk fuels, it's not going to happen both because of price but also because of scale. So it's the other vast array of chemicals that we get from fossil fuel refining where I think synthetic biology can have an impact on that industry. The other places, I think, in terms of decarbonisation, I think synthetic biology has a lot of potential. I talked about one of the companies working on methane reduction in cattle, but I think there's a variety of other stuff, possibilities in decarbonisation and carbon negative biomanufacturing where synthetic biology can have an impact. But yeah, unless the price of fossil fuels goes up by 10-fold, 100-fold, we're not going to be producing bulk fuel replacements with microbial fermentation.
Fascinating. I'm a mucosal immunologist, and my interest is controlling human microbiomes through driving a mucosal immune response. Cool. And in doing that, we use non-type Haemophilus influenzae. And our big issue is fermentation in volume. And it seems, and this must be an issue that, looking at many of the things you're doing, is availability of high volume, large fermentation facilities. And we're starting to look outside of Australia. And I'm just wondering how your organisation is addressing this, because we'd love to have one in Sydney.
Yes, so what are we doing to address this? We're trying to lobby state and federal governments to... So there's a massive fermentation shortfall everywhere in the world. Probably the only country that has enough fermentation is Denmark. And particularly when you're doing something that's a first in the world plant. No one's ever built a plant to produce, let's say, I don't know, the particular oil or whatever. If you're a startup company, you can't go to venture, it's going to cost $200 million to build a plant. You can't go to venture capital to build a plant to get that scale of money. But you can't go to a bank because a bank's like, prove to us that it works. And yes, you've done it at scale. So if you've built 4 factories and whatever, it's very easy to go to a bank and get a loan to build the 5th one. But to build your first of its kind facility, very hard to get money out of the bank. And this is a problem not just in Australia, but in the US and elsewhere. And so people are exploring various creative schemes, but my belief is that is that you need some degree of government underwriting to de-risk these investments for large scale. And what I think would be, so what we don't want is government building like one large fermentation plant and dumping it, I don't know, in some random rural town in New South Wales, because that's going to turn out to be a white elephant. What I think we need to do is have some sort of, industry needs to have skin in the game. So if a company is willing to put up 50% of the cost, then I think if we had a scheme where the government would back it with the other 50%, and I'm aware of the future fund and things like that are possibilities in that sort of space, but I think I think what we really need is a dedicated government scheme to de-risk investment in large-scale infrastructure. The US have something called Biomaid, where it's actually funded by the Department of Defence, but they're not making defence-related goods. It's actually because they see synthetic biology and biomanufacturing as a key strategic capability. They've put this scheme up, and it's not really for building full-scale plants, but it's for building proof of concept or scale-up plant. But they have this scheme where it requires industry, government, and academic partnership, and basically it's 50-50 funded by industry and government. So I think we need something analogous to that.
Questions? Tibor?
Thank you. Thanks, Ian. That's fabulous. About 3 billion years ago, the atmosphere was 35% carbon dioxide. It was 350,000 parts per million. People don't know that. They think that 400 parts per million is novel, is new. Along came photosynthesis, and it took nature approximately 150 to 200 million years to get the carbon dioxide out of the atmosphere and put it away under the ground in the form of oil and coal and gas. And we're very grateful that it does. Of course, it's taking us about 200 years to get it all out again and put it back in the atmosphere.
Yes.
So we're really much more efficient than nature. So
more destructive anyway.
Exactly. So my question is, there are two things that we can do to help us with addressing climate change. We're looking at several degrees of warming over the next century or two. There's no question about that anymore, I don't think. We can either improve photosynthesis or find something which is like photosynthesis but quicker and faster and better and more productive or more hungry for carbon dioxide or something. Or we can, I don't know, or the other thing we can do is find some way of stopping from photosynthesis from breaking. One of the problems we have is that if wet bulb temperature goes above 35 degrees, photosynthesis stops working. There's C3 and C4, you know all about that. And photosynthesis degrades very rapidly as temperature rises. And we're now close to where that's about to happen. So, two-pronged question. One of them, could we do anything to save photosynthesis? And second, do you have alternatives in your plan to find things that eat photosynthesis and will help us along in cleaning up the mess?
So, yeah, good question. I don't have easy answers. Various research groups around the world have been, working for years, decades to try and improve photosynthesis. I think that's a very hard, non-trivial problem, even with that modern synthetic biology. One low-hanging fruit where I think we can have an impact is methane production. And so there are various methane, a lot of human activities produce, so methane is 10 times or whatever, no, I'm not a chemist, many times more power, more heat.
28.
28, thank you. I said I'm not a chemist. Excellent. Good. I'm always happy to be corrected by people who actually know stuff. Where was I? But it's there for a much shorter period. It's not there for thousands of years. It's there for tens or hundreds of years. 12 years. Thank you. Facts, always a helpful thing. But the methane is in, I'm going to say, an equilibrium situation, and we're pushing hard on basically ways that increase that methane production through a variety of human activities. But one of the main drivers is actually changes in the microbiome composition of soils. And in the future, one of the main problems is going to be release of methane clathrates from thawing tundra and things like that. There are a number of companies working on things that might be strategies to reduce methane production from both agriculture and soils. And I think that's an area in which synthetic biology and microbial manipulation can have a real impact. If you do a back of the envelope calculation, we could, if there was the money and political will and the engineering know-how, which is not my area of expertise, but a back of the envelope calculation, if you made, I'm going to say, 1,000-kilometre raceway and cycled salty water through it and grew cyanobacteria over it, you could actually, and collected that cyanobacteria intermittently and turned it into biochar or whatever and buried that in the soil, you can conceivably have a drawdown impact on us. But the investment that would need to do that, there's not currently the political will to make that happen, and it's easy for me to do a back of the envelope calculation and say that might work. I'm not an engineer. Whether that would actually work in principle, in practice, I can't answer that question. So climate change is both a scientific and an engineering, but also a political challenge, I guess, is what I'm saying. Yeah.
And you mentioned defence, and there are always downsides to every technology. So what are the nasties that could come out of synthetic biology that we should at least contemplate?
I guess the number one potential one is engineering pathogens to be more pathogenic, or engineering them to attack crops or other things like that. So there's certainly areas that I know are on watch lists of AUKUS, and I'm sure most militaries on the planet. The good news is most of the time when we engineer microbes, we make them more feeble and less able to live in the real world. And we see that with our industrial microbes. So I think so the final stage of getting a centre of excellence is you have an interview panel in Canberra, and I think at that interview panel I got asked 3 different ways. How do we know, Ian, that you're not going to destroy the world with your synthetic microbes? And we can talk about biosafety regulation, but the real answer is most of the time when we make change. So organisms have been living out in the real world for 3.8 billion years each, and competing with each other for resources and whatever, and are finally tuned to survive in their particular. And so usually when we take an organism and change it and put it in the real world, it goes, oh my God, I can't handle this. We're good at growing things in big tanks where there's nothing else. And so I guess I can't guarantee there's not significant defence threats from engineered organisms, but I'm mostly reassured by that I think most people trying to engineer organisms are actually going to make them less effective rather than more effective as a pathogen.
Another big picture question, I can see Bryn's got one, another chemist, so watch out. Is there any point to doing any synthetic biology experiments in space? And if there is, what is being done currently?
So there is. We actually wrote a concept paper, opinion piece, I think in Nature Communications or maybe Nature Foods, I don't remember now. A year or 2 ago, about the possibility. So one low-hanging fruit is basically using it as a nutrition and almost like a pharmacy for astronauts or for colonists to another planet. This is all very sci-fi, so I think practical. Practical utility in the short term, I'm going to say close to zero. But yes, if Elon Musk wants to give me $1 billion to engineer microbes to live on Mars, I'm happy to take the $1 billion. Well, actually, I don't know if I am happy in the current climate, but yeah. No, I said 1 billion. Yeah. Where was I? Yes. So, you could conceivably engineer microbes for production of, vitamins or other things that, astronauts might get deficient on over time. And in this opinion piece, we said you can conceivably 3D print them into things that resemble food and engineer, other microbes to give it texture that resemble. It's all a bit science fiction, and I think not a short-term practical reality, but not inconceivable in the fullness of time. But is this an area where I think synthetic biology has the most promise and impact? No, I'd rather see us actually trying to solve problems on Earth rather than in space.
Bryn.
So, thank you. I am a chemist. Last year's Nobel Prize for Chemistry was won by the developers of AlphaFold. So now you can work out the structure of any protein that you may want. How's that affecting your research?
It's very helpful because we can now predict the structure of any protein we want. It's the other, actually more excited by Baker's more recent research. So they're now had some successes. It's very hard, but they're able to do de novo design. I don't know if that's too technical a word. They can design things that don't exist in nature on a computer and build the protein, and maybe 1 to 1,000 times it actually works. And so that opens up exciting possibilities of building enzymes that can do things that no natural protein can do. They can bind, and so I think the example paper I can think of from the Baker group was they designed a anti-venom for a particular snake toxin by just designing on a computer, building it, looked nothing like any natural protein, and it worked to bind this snake toxin. So you can conceive of scenarios now where, if we want to build microbes that sequester rare earths or other things that biology traditionally it's hard for, but we would have use for, then this offers the possibility of doing that. But it's not yet a routine, I'll just push a button and dial up the protein I want. It's still, it's hard. It's, trial and error. It's, here's 1,000 options and look, one of them works, sort of thing. So we're not good at it yet, but this is also our first baby steps. Give it another few years and, maybe we are then routinely designing novel proteins on a computer that can do stuff.
Tweak the ones you've got.
Yes. The other, what I would like to see, or what I would like to see AI do, and we currently, so at the moment we have this design, build, test, learn cycle. And with the robots, we can automate the building and the testing. If we had good AI, we could conceivably automate the design. So I could I dream of a scenario where I come to my AI master and say, give me 1,000 designs of, a yeast strain that can, eat methanol and turn it into aviation fuel. And then tell my robots, build these 1,000 designs. Oh, look, this one actually worked. We don't have that. And, but, 5 years ago, I would have said, it's a, we're not going to have, AI capable, I fell out of my chair, when the first papers came out on, AI structural prediction of proteins. So, maybe another 5 years, maybe in another. I'm not an AI person; it's not my expertise. I talk to AI people, but it's challenging because they speak in their jargon, I speak in my jargon, and communication is a challenge. That's where I see exciting possibilities for synthetic biology. But AlphaFold and RosettaFold and these, protein structure prediction programs are revolutionising a corner of biology.
One last question. Thanking you. That's what I'm doing. This is a very challenging to thank you. It's really quite an extraordinary talk. I feel as though we've got the sort of Renaissance man for the 21st century. from climate change, microbiomes, these black soldier flies, startups, and a whole new world which wasn't there. I've just been reading Janet Brown's account of Darwin's voyage on the Beagle and his early life, and what's striking about that is Darwin's excitement and enthusiasm and range of things that he's interested in, and throughout the voyage, because he wrote letters all the time. And what I think we've had tonight from you is a sort of sense of a world as different as Darwin's world, as Darwin showed us from the Beagle. No. I mean, it's the difference. It's the complete. I mean, I know some of you know it, but I don't know it. It's really quite extraordinary. But also the commitment to letting scientific reasoning take us where we will. Sally, who organised this event, was summoned away, as she said, to the Blue Mountains. But she wrote to me saying that she'd hoped to have as the plan for this year of lectures that she's doing, a series which got to the value of science and civil society in addressing global challenges. And she says your groundbreaking research in synthetic biology does exactly this. It's got the potential to address sustainable development goals such as clean fuel, zero hunger, and reduce inequality. It's an extraordinary area, and it is quite different, and I know many of the more scientifically expert people might well know more about it, but for me it's been riveting and quite, those funny little things edging across the screen. I think, oh dear. So I think what you've done is opened the year doing exactly what Sally wanted, and I know she's incredibly disappointed not to be here. We at the Royal Society of New South Wales, we're trying to mobilise multidisciplinary expertise to address national and global challenges. And that means reasoned evidence across boundaries of all sorts, disciplinary, political, and others. And we're talking about science here in this broader sense, as you were when you were talking about your sociologists and your musicians. Well, you're a bit rude about music, but...
Well, we have a creative in residence who is and our creative in residence this year is a musician.
I totally approve. I think that's really great. So getting all of these to go together, and we're trying to bring together academics, but also industry, government, civil society, those people who are involved in the whole economic structures of our society, and see if we can start to see a way through. And I think what I wanted to finish with was what I thought was probably a really good motto for our year. You said you're always happy to be corrected by people who know stuff. And that's a really good thing for us to think about, I think, in the society. Be corrected by people who know stuff. So thank you very much, Ian.
Well, thank you. That's lovely. Thank you.